my new style of microphone. Attaching it to the front of your face. So, good morning, intrepid learners. Neither gloom nor rain nor head colds can keep us from our appointed rounds. We are back today welcoming Professor Sandquist back for the third of this lecture's You and Your Drugs. And I also want to tell you that the next series will be by Professor Emeritus Bruce Boyles on November 6th and 13th. He's talking about influenza A virus. Timely topic. And registration is open online, or see Judy Hunter at the registration desk if you want to talk more about that. Okay, the usual reminders. If you're using a tea coil, time to turn it on. Have you turned your cell phone off or down? I haven't. How about off? Because you can always make calls over break. And the way we've been doing it the last couple weeks is that when you have a question, you just hold your hand up. Josh is going to recognize you. You'll ask your question, and he'll repeat it into the mic, and that seems to have been working pretty well. Remember that the two books that he recommended, Powerful Medicine, The Benefits, Risks, and Costs of Prescription Drugs, that one is, a copy was available at the library if you want to get on the list. And the library and bookstore have also ordered the other book called Bitter Roots. Josh, welcome back.
Thanks, Janet. Let me check the microphone again. Is it okay in the back? Good. All right. Thank you all for coming back again today. We're going to talk about a variety of things today. Hopefully they all come together in the end. It may seem kind of like a disparate mix for a while, but hopefully it'll all make sense at the end, and hopefully I don't go too fast. So we'll go ahead and jump right in. I'm going to start with a quick review of Lecture 2, which was a lot about the science, how drugs work, get into your body and work. There's a bit about metabolism, genetics that I didn't quite get to on last week, so I want to cover that bit. And then today we're going to start off with a big focus on drug development. How are drugs discovered, how are they developed, how are they made, so we can understand a little bit more of the costs and benefits of that. A little bit of a public service announcement on over-the-counter medicines. And then hopefully there's time to wrap this all together in the story of aspirin, anti-inflammatory drugs, and troubles in big pharma. So we'll see how this all works. All right, so Lecture 2, we talked about how drugs have to work by physically getting into your body and binding to some specific, usually a protein, some specific targets that they get there, get to that site, and physically interact with the protein. And drugs are designed in such a way as possible that they specifically bind one thing and only one thing. That doesn't always work out the best, but that's the goal. So the first, since most of the drugs we take are orally in pill form, that's what we focused on. And the first major hurdle then is just getting into the body, getting through those cells in the intestine into the body. From there it gets into the blood, and the blood moves it all around the body. To reach its target, the first place the blood goes after leaving the intestine is through the liver, where a lot of things are metabolized. So we have to factor that in. And metabolism is necessary for many drugs to change them so that the body is able to remove them. And we talked about how because there's a relatively small number of enzymes that break down most of the drugs, there's a potential there for interactions. So if two drugs are broken down by the same enzyme, having two drugs on board may slow down the breakdown of one or the other, which could cause a problem. So it means maybe the drug lasts longer in the body or gets to a higher concentration than we normally want. So we got to there, and I was also going to talk about how metabolism enzymes can be dramatically different based on people's genetics. So that's what we're going to pick up today. All right, so we looked at this diagram last week, and this is sort of just another good review. So we talked about how you take a pill, it comes through the stomach, goes to the small intestine, and here's blowing up that small intestine here, and the drug has to be absorbed through the membrane into the bloodstream. And there are some enzymes, these metabolism enzymes, that live right there in the intestine. So there's some metabolism occurring on the way into the body even. And we talk about how grapefruit juice can create problems here. So it goes into the blood, goes to the liver, more metabolism, and then eventually it can go to the heart and move around the body. All right, so we talked about how if 2 drugs were metabolized by this guy, that could create problems, or if you have grapefruit juice, could affect the metabolism of that drug. But what I didn't quite get to was how genetics can play an important role in metabolism. So here we have 2 graphs looking at what's called the half-life. So this is basically the amount of time it takes for the body to remove half the drug. Okay. So in this example here, it takes almost 12 hours for this patient to remove half this drug, antipyrine, which is an analgesic, a pain medication. And this drug, antipyrine, is exclusively metabolized by these CYP enzymes we talked about. So people were studying the impact of genetics on this, and they have here A, B, C, D, E, F, G, H, I number of pairs of twins, and you have sort of red and pinkish showing the rate at which each twin metabolizes antipyrine. So if you look at identical twins who have the same genetics, there's variation from twin set to twin set. So twin set F is slow, because it takes almost 15 hours to get rid of half the drug. Twin set I is relatively fast, because it takes only 6 hours to get rid of half the drug. But within the twins, they're very similar. Now if we go look at fraternal twins, so they have the same parents, just they're not genetically identical, we see much more variation. There's variation between A and B and A and G, but also even within A, the 2 twins have dramatic differences. So this shows that a person's genetics can affect their metabolism of their drugs. Here's another example of this, just to show how that can play out in the way the drug works. Mercaptopurine is an immunosuppressive that's often used in leukemia. It's metabolized by an enzyme we're just going to call TPMT. Now there are different versions of this TPMT enzyme that are just natural. Different people will have different versions. 3 or 4 maybe different versions. And so just a quick review, everyone has 2 copies of a gene for each gene. So you get 1 copy from your mom and 1 copy from your dad. And so, let's back up here. So there could be TPMT version 1, TPMT version 2, there's 3A, 3C. What we're looking at on this axis here is the activity, so how functional, how active is this enzyme. The 1 version, 1/1 means both copies of your TPMT gene, copy got from mom and from dad, are both version 1. In that case, we see there's some variation, but they're all pretty fast-moving enzymes. Now let's say you're someone who actually got a version 2 of this enzyme from both mom and dad. This is a very inactive enzyme, very, very little activity. Or if you're a person who got version 1 from, let's say, dad and version 2 from mom, you're right in the middle. So there's a lot of variation in how much people can metabolize this drug. Now if we take these groups and we ask, how long does it take for the doctor to have to change their dose? This is often something, well, they start with a dose, and they come back and make sure things are working. Immunosuppressives work by suppressing your immune system, which is important in leukemia. But if you suppress your immune system too much, you're just going to get sick, or really sick, from very common things, just like a cold. So we don't want this being too strong of an effect. So they very carefully monitor the dose. Now, if you're someone in the orange is the wild type, that means you have 2 copies of the one, the fast version. Oh gosh, what'd I do here? All right. Over time, in years, the percent of people who have to get their dosage decreased is really small, maybe 20%. So they're very stable. That's the wild type. The green, homozygous variant, don't worry about what that means. That just means you have both copies of number 2. After 1 month, 50% of the people had to decrease their dose because their enzyme is much slower here, and there's a lot more drug in the system than we expect. After 6 months, 100 people had to change their dose. So these genetic differences in people have a serious effect on how the drug works, and the doctors have to monitor them differently. So it'd be great, if we had a test where someone could come in and we could quickly say, oh, you have version 1 and version 2, and we could know how to approach your treatment. Well, that's sort of the goal or the promise of pharmacogenetics. Personalized medicine. We could look at your genes and say, this is we shouldn't use this drug in you, or we should, but we have to use it in this way. That's sort of the promise of pharmacogenetics. Which sounds great, but we're not really there yet. So the problem is, in most cases, there's not really a specific genetic test available. We know there's differences in people that are probably genetic. The example that I showed with the identical versus the fraternal twins, that's proof that there's a genetic difference. That doesn't necessarily say what the enzyme is or what the gene is. So we know there's a genetic difference, but we don't know how to test for it ahead of time. Or even if there is a specific genetic test, often they're expensive or insurance companies won't cover it. So it's not just possible for you to come in, the doctor to swab your cheek cells, scan your DNA and say, you have this genetic phenotype. But because of this promise, and because people are observant, over time, people made observations like, I noticed that my black patients were responding in this way to a drug differently than my white patients. And this prompted people to do studies to test those sort of things. Do black people respond differently than white people to this drug? So what happened is, in the absence of specific genetic tests, and this was even happening decades and decades ago before we even had really genetic screening, people were using skin color and other superficial characteristics as sort of a proxy for genetic ancestry, thinking if you had similar genetic ancestry, you would have predictive effects of these things. The problem is that presumed racial identity is a very poor indicator of genetic ancestry. What one person thinks of as black may be different. Someone who's African American in origin, or maybe a native Brazilian, both have dark skin, but very different genetic backgrounds. Plus, race just isn't a genetically encoded thing, despite the fact that many of us believe that way. Race is more of a social construct. As an example of this, there was a study that they looked at where they showed that black African American people were responding poorly to these hypertensive drugs. And so they thought, well, black people, you're dark skinned, you're one race, you're different than white, and so you're responding differently based on your genetics. Well, the problem with this is that for social reasons, black people, particularly in a lot of larger cities, are typically poorer than white people. They have different diets, and it turns out that when you're poor, you buy cheaper food, a lot more sodium. They had higher sodium levels in their blood, which affected the heart, the blood pressure medication. So that was a complete social difference that was causing that difference. It wasn't the fact that they were black and they had different genes, it was the fact that they were black and they were in a racist society that caused them to have a different environment. So what happens is, there was the best of intentions. Let's try to use whatever data we have available as a window into genetics so we can tailor our medicines, trying to get at this goal of personalized medicines before we had the ability to screen genes. The problem is it ended up having sort of this perverse backwards effect where we took individuals and bent them into these generic categories of black-white, which had no predictive power. It's also controversial because using this, well, again, while well-intentioned, sort of promotes the idea in society that race has a genetic basis, which it doesn't, so it makes it harder for us to get past that problem. So, long story short, while there's a lot of promise in genetics and their ability to be used for medicine, we just have a long ways to go yet to where we can really see the promise of that. Okay, so that was part of lecture 1. We're going to move on to a different idea, and we'll come back to some of these things in a little bit. So, drug discovery and development. You've probably all heard that drug discovery and development takes a long time and it's really expensive. That's just sort of these things that we bemoan, right? And here's a chart that we'll come back to over time here, looking at kind of showing you the drug discovery process really quick hand. The point I want to make right this instant is that to develop a drug from when you're sort of the initial discovery to where it's on the market can take 10 to 15 years and cost billions of dollars. So I looked at this study done by Tufts. This is a few years old now. I think it was published maybe 8 or 9 years ago. And they looked at the preceding 15 years, adding up the costs of drug production. And over that 15-year period, they found that on average, it takes about $350 million to develop a single drug. So the cost going in to make that drug. But many drugs for a drug company actually fail. They'll spend the $350 million and they don't get anything they can use out of it. So when you factor in how many fail, the average is about $5 billion cost per drug that's actually on the market. So this is a ton of money that goes into making a single drug. So let's unpack this a little bit. Why does it cost so much? Why does it take so much time to make a drug? There's sort of 2 broad phases. We'll talk about discovery, where you're trying to find molecules that will actually work as a drug. You find one that has promise, that's the discovery phase. And then there's the development phase, where you have to take that drug that has promise and fiddle with it and convert it and make it to where it can actually be used by people out in society. That's the development phase. So let's start with thinking about drug discovery. So obviously you need to start with some sort of disease or condition that you want to treat. Asthma, Alzheimer's, cancer, whatever the condition is. And then a lot of research has to go into figuring out the mechanism. So remember, we said drugs only work by binding a very specific protein, and they can only increase or decrease normal functions in the body. They can't add new functions. So there's a lot of work that has to go into basically finding what's the one protein that I can have a drug bind to and cause the effect that I want. So we have to understand what's going wrong in the disease in the first place. So eventually, a molecular target, so a single molecule, a single protein in the body will be found that, oh, this is what's going wrong in this disease. Or maybe we'll discover we don't know what's going wrong, but we know if we change this protein's function, that can compensate for what's going wrong. We don't always know what's going wrong. We have to find some protein that we think will, if we target it, it will produce the effect we want. And you verify that that indeed works that way. Then you have to identify potential lead compounds, drugs that might work here. I want to point out that all this work and all this research and understanding the disease and finding a potential target for a drug, some drug companies do that research themselves, but a lot of this is actually done in research universities, research institutions that are funded by federal money, by public money. So we'll come back to this a bit later. Just keep in mind, a lot of this initial work that goes into is actually publicly funded research. Okay, so we found a target. Now we have to find chemicals that we think will bind this target and do something. There's a variety of ways this can happen. So maybe there'll be natural compounds that people have discovered. maybe, you know, aspirin is something we're going to talk about more later. That comes from the bark of the willow tree. Penicillin is from a mold. Morphine is from the poppies. People were using that long years ago as an illicit, well, it probably wasn't illicit then, but it's an illicit drug now. So these are all things that sort of discovered by accident had these properties, and then we fine-tune them into a drug we can use. L-Dopa is used for Parkinson's. That's just a slightly altered version of dopamine, which already exists in our body. So these are all drugs that were found somehow because they already existed in nature, and then we just bent them to pharmaceutical purposes. Other ways of finding lead compounds, I think I touched on this the other day, is this screening of chemical libraries. So pharmaceutical companies will have freezers full of tens of thousands of different chemicals. And when they find that they have a target that they're interested in, they'll set up some sort of assay where they can use robotics to kind of quickly screen through those 10,000 chemicals and see if any have any promise. So this is just sort of a random shotgun approach. They're just throwing stuff at it, hopefully finding some that will do something promising. There's not real much rationality behind that. There's other approaches. The chemists will work and think about things and cogitate and make changes to drugs. Some people are using biotechnology, like bacteria, to try to design chemicals. There's a variety of different ways. Here's an example I wanted to give of some of the optimization that's done on these chemicals. So let's say this is your lead compound. This was the compound that was found to maybe have some promise. People will step in and start making very minor changes to it and see what that does. So in this case, they change, I don't know if you can read this here, it's kind of fuzzy. There's a chlorine up here, and they changed it to an oxygen with a carbon on it. And they wanted to measure, this number here is a measure of how well it binds the target. So larger number is worse, smaller number is better. Not really important, but they say, oh, when we changed this, it didn't have much of an effect, but when we changed this hydrogen to this group over here, it actually binds a lot better. So you find something, maybe through your screen, or it was a natural compound, and then chemists go to work making fine changes to it, seeing if they can improve it some way. So that's all this discovery. That sounds like a lot of work already, and that's just finding chemicals that maybe might work. Okay? We have no idea if it's going to work in the body yet. So we'll maybe narrow it down from thousands of chemicals that we're starting with, and if you're lucky, you'll have maybe a couple hundred potential compounds that might be useful. So now we get to these preclinical studies. So far, none of these chemicals have been used in people, but that's the ultimate goal. So we have these compounds that we know will bind a protein and do something, but is it going to kill the person? Is it going to work in the person? That's what the clinical trials are for. So the preclinical trials are the step into that. So the first thing in the preclinical trials, they want to test to see if this compound will be safe in humans. So they'll be maybe using human cells or animal studies to see if it looks like it's going to be severely toxic right away. You can at least get rid of those. So they're determining its safety profile. If this is going to be used in clinical studies, you need a large amount of it. Maybe you just have a small amount to do those initial tests. Well, now chemists have to try to figure out how can we scale up and make more of this to have enough to do our studies with. So maybe by the time you get through these, something that's able to be scaled up easily and cost effectively, something that's not toxic in animals, you go from maybe those initial hundreds down to a handful of chemicals that you might actually use in people in clinical trials. So now you take your 5 drugs, you file an IND, which is an investigative new drug, the FDA saying, I have this new drug I want to use in clinical trials, will you let me do it? Hopefully they look at these preclinical studies, say, yeah, that's not too toxic in animals, or whatever they used it to decide, and say, okay, you can do clinical trials with these compounds. Then you've probably heard these phrases before, phase 1, phase 2, phase 3 clinical trials. There's a whole series of trials that they go through in people. So after they've submitted their investigational new drug and it gets approved, we're slowly working this in carefully by baby steps to the point where we can use it in people, a large number of people. The first step, the phase 1, we're being really cautious. We're giving this drug to a small number of people, and these aren't people that have the condition that we're trying to treat. These are just healthy volunteers. Usually young, 20-something, physically fit people. The goal here is just to sort of understand how the drug works in the body. So remember we talked about the time it takes to get in, the time it takes to get out, how quickly is it metabolized. These are things we don't really know from all those previous studies. So they find healthy volunteers to take these, so we can just measure those parameters. So we get better understanding of how much we need to give, how fast it works. If it makes it through phase 1, we go to phase 2. Now you're actually looking, okay, we know how it works in the body. Let's see if it actually produces the desired effect. Does it treat the thing that we want it to treat? So now you get a larger number of people. These are actual patients that have the condition. Asking questions like, is it effective? Does it beat the placebo? Are there major side effects? Does it work like how we thought it would work? What sort of doses do we need to actually see the effect we want? So these are things we're figuring out. Oops. Finally, phase 3, the last one before it goes on the market. Up until this point, it's maybe only been in a couple hundred people. If there's a really serious side effect, like death or something, which is possible, but there's other serious side effects. Let's say that happens in 0.1% of people. You may have never seen it in only 500 people, because that would be like half a person. So you can't kill half a person. So this is something, they scale it up. Basically, similar studies as before, they're using it like they want to use it in the market, determine the overall risk-benefit ratio. They decide how they're going to label it for doctors to use. These are the costliest and longest trials, because now you're using a couple thousand people. You have to use it for longer periods of time to make sure there aren't side effects that are going to creep up. So some considerations for these trials. Some of these things should look at, if we think back to the first lecture where we talked about estrogen and how estrogen was going to be used, and we thought they were going to use it for all these things that would be useful, but they didn't really do the proper type of studies to prove that it'd be effective. That's where all these sort of considerations come in. So when you're doing your clinical trial, are you going to compare it to placebo? Or maybe you're going to compare it to some standard treatment. There's something that's already used for that. We want to see if our new drug does better than that thing that's already used, or at least as well as that thing that's already on the market. We want to randomize people. So that was the estrogen thing, where we had people who are already taking estrogen. Well, that's one type of group because they were very proactive about their health, and then people who weren't taking estrogen. So that was sort of biased in a way. So we can't have that bias in there. We talked about double blind. Neither the patient nor the doctor knows that they're taking the drug. Crossover studies are where it's this blind situation. You don't know what you're taking, but they change your medication halfway through. You switch from maybe the placebo into the medication. This is, again, one extra level of proof that the drug is actually doing something. If you're taking it and you switch to placebo and you start to get worse, then they know the drug was actually doing something, right? The last thing to consider is a little bit tricky. What are you going to use as a marker or a surrogate endpoint? Let's say you're trying to develop a drug that's going to reduce the risk of heart attacks. It's hard to prove that in a year-long study, because you never know when you're going to have a heart attack, and that's a really long endpoint. You can't do the study until 50 people have heart attacks and decide did it make it better. So they need some other marker. What's something that we can measure in the short term that's really predictive of heart attacks? This was the problem again with the estrogen, the hormone replacement. Again, they couldn't show that the hormone estrogen replacements were preventing heart attacks in postmenopausal women, but they showed that, it was reducing cholesterol, and they were using cholesterol as a marker for heart attacks, because we know high cholesterol is often seen in people who have heart attacks, but it's not a predictive marker. So again, these are all the sort of problems that now we know we try to avoid that were happening back in the 70s when people thought we should use estrogen in all these ways. So finally, we get through all that. If you're lucky, you have one drug that made it all the way through. You can file a new drug application with the FDA and say, I have this new drug that I want to sell on the market. Maybe you did all that and none of those meet all those criteria. That's why drugs cost $5 billion to make. But even after you get it on the market, you still have to find, maybe your chemists are still working overtime. Now this is going to be in 1 million people. We have to figure out a way to make this cheaply. And if after it gets on the market, the drug companies still have to follow up with what's called phase 4. They have to keep track of things. If it's something like a 0.1% side effect, again, that may not have been very common in maybe the 5,000 people who have been exposed to this drug. When it goes to 1 million people, 0.1% is now actually a large number of people. So we have to keep track of these things, look at the long-term safety, are different subgroups of patients being affected differently than others. So there's a lot of work that's still being done after the drug goes on the market. So that's the general process. And maybe this raised some questions for you, so this is where I want to take a point. We'll just break this down and think about this in terms of society. Yeah?
What point in the drug development process would the drug be patented?
That's a good question. We're going to talk about patents in a little bit. But often the chemicals are patented maybe even before the drug discovery processes begin. They'll have the chemical, they've made it, they want to patent it, assuming it may be valuable someday. So they probably want to wait as long as possible to patent it, because the patent does expire, usually after 20 years in the United States. That's sort of a game they play. If they're worried someone else might come in and patent it before them, they have to patent it. Yes?
Aren't there some dangers to young, healthy people who are taking drugs for a condition they don't have?
Yes, the question is, aren't there some dangers for young, healthy people in phase 1 taking these drugs for a condition they don't have? And that is one of the points here I wanted to make. Guess my question here on the ethics here.
Sorry.
No, that's fine, that's good. This is exactly what I want. You guys are a great audience, you're anticipating where I'm going. So who signs up for these trials? Well, oftentimes you're given some small fee, maybe a couple $100s or something to participate in the trial. So it typically ends up being poor people, people who could use that extra couple $100s. So this is the ethical dilemma. We have poorer people in our society sort of subsidizing a gain that everyone else in society gets, potential new medicine. But that burden is disproportionately carried by poor people. So that's an ethical problem. I don't know if anyone really has a good solution for that. Same idea, if you want to tell people, okay, you're signing up for this study, there's this concept of informed consent. You can only agree to participate in this if you feel fully informed about the potential risks. Someone can sit and read 10 pages of science and not really be informed if it's not written in a way that they can understand it. So what does informed consent really look like? These are related questions of who participates in these trials. I saw another hand, yeah.
Yeah, how systematic is that last phase, the phase where it's being sold to the public?
So the question is, how systematic is that phase 4 where now it's on the market, but companies have to keep track of adverse event? Well, the way, this is a little bit out of my knowledge realm here, but what I understand of it is the drug companies have sort of these databases set up, and people who prescribe and give medicines know where they are. So if they are seeing an adverse effect, they can go to that database and say, I saw this effect on this drug with this patient. And the drug company has to monitor that and look for any patterns. Oh, we're seeing all over the country that this group is having problems with our drug. So that kind of goes on indefinitely, as far as I know. They're keeping track of potential long-term effects of these through these databases where everyone knows to try to put in information when they see something. Does that answer your?
Well, when you use the term everyone, you're talking about doctors, not patients.
Yes, but by everyone I mean doctors. So typically if the patient's saying, oh, this is really making me have a bad headache or nauseous all the time, then the doctor's job is to note that, and then report that to the appropriate place so the drug companies can look and see, is that a weird thing that only happens in one person, or is that something that's common across the country? Yes?
At what point and who decides the list of side effects on a drug?
So the question is, at what point and who decides the list of side effects? So these are things that are noticed during the clinical trials. When the company puts in an application for a new drug, they have to show the FDA the data. We noticed, 5% of patients taking this drug had headaches. So they list all those things in their report, and then the FDA, that's part of their decision. They decide, well, this drug, considering its benefits, it's okay that 5% of people have headaches and 5% of people are nauseous. So that's part of what the FDA does. That has to be monitored over time. So if a new side effect comes up that they didn't notice in the clinical trials, maybe heart attack happens in 0.1%, which is a small number that they never really noticed in the clinical trials. But now that it's in 1 million people, that's a noticeable number. They have to update that. Then the FDA may decide, well, if this is just a medicine you're taking for like a headache and it's causing heart attacks, maybe that's not an acceptable risk. But if it's a medicine for cancer and it's causing heart attacks in a really small number of people, well, maybe we'll leave it on the market, and then it's the doctor's job to decide with the patient if that's an acceptable risk. So it continues to change over time.
Are they listed in any particular order?
That is a good question. The question is, are the side effects listed in any particular order? I don't know. It makes sense if it was most common to least common. That's a good question. I should know that, but I don't know the answer to that. So, some other ethical concerns about the drug development process. Should drugs for certain diseases be fast-tracked? We've had some Ebola scares the past few years. You can imagine an outbreak happening in the US next year. We don't want to wait 15 years to develop a drug that can be useful on Ebola. This was really important with AIDS in the 80s and 90s. There were drugs that were sort of fast-tracked through this process. The hurdle to consider it safe was lowered just a little bit, to take a little bit less time to get it to the market. The rationale being this is a very serious condition. We're willing to have there be a few mistakes, or have there be maybe a drug that's not quite perfect in order to try to treat this serious condition. So some drugs will be fast-tracked. They still have to be monitored, but they're allowed to get through the process a little bit faster. Who's responsible to develop drugs that don't make money? So antibiotics. We have a lot of cheap antibiotics right now in the market. It's not worth it for a drug company to spend $5 billion making a really expensive antibiotic they're not going to be able to sell, because they'll be able to buy another one that's really cheap. But we know that maybe in 20 years, none of these antibiotics are going to work anymore. So whose responsibility is it to pay for that now to make the drugs that we're going to need in 20 years if they're not going to make money for that drug right now? This is a problem I sort of alluded to when I talked about the public funding for the drug discovery. There's these academia-industry partnerships. Public money is being used to study diseases that's converted into financial gains for private companies, because they didn't have to do the work to discover the target, but now they can capitalize on it. Maybe some people consider that public investment worthwhile, because it's not really profitable for companies to spend all that money, because they're kind of shooting blind. So maybe that's an acceptable outcome about a public investment. That's debatable. You have this, one question is, but now you have this public knowledge. So they identify this disease target. Now the drug companies are trying to do some private things with it that they don't want to share because they don't want some other drug company to steal their. So again, I mean, it's the same idea, public investment, public knowledge versus private proprietary ownership of these things. These are some questions that our society's dealing with because of the way drugs are developed in our country. Ghost authorship, I don't know why I kept this in there, sort of this niche complaint of mine, but there are drug companies that will do research into a disease or a drug. So they're paid by the drug company. They'll kind of write up a report on it, and then they'll have an academic advisor that they pay. And the academic advisor didn't do the research. Their name will go on the published article, sort of lending their cachet to it. This is an authority. And then the drug company will keep their researchers' names off the paper. This is an acceptable, so it kind of hides some of these conflict of interests. I think I have one more point and then we'll, drug patenting. So it's almost break time, so I'll take a question, we'll come back to do drug patenting, and then we'll move on to some new ideas. Yeah?
I need you to tweak my understanding, the public money basis for private gains. Public money goes in, some public money goes into university research.
Mm-hmm.
Am I hearing you say that public money then can not be shared widely? It can be captured by a particular private group just for their own use?
Okay, so the question was to explain what I meant here by public money being used for private company gain. And what I mean here is, let's take a complicated disease like Alzheimer's, where we don't maybe fully understand the disease. So there are people who work at universities getting grants from the federal government, which is taxpayer money, that funds their research. Now let's say a researcher at University of Iowa discovers, oh, this protein is essential to Alzheimer's. All that work, that expense to identify that target was done with taxpayer money. Now a drug company who reads that paper can come in and develop a drug against that target and then sell that drug for billions of dollars. So the drug company's gains, financial gains from selling that drug were based on work that was done by taxpayer money.
Okay, but that research that is done can be available to others. That's not just captured by one particular association.
Yeah, so the question is about the public research being done, being available to everyone. Yes, the people who do that work with the taxpayer money, they publish their paper in open journals. Any drug company could read that, but the drug company didn't have to do all that expense to find that target. And then now you have a patient who's going to have Alzheimer's and is going to have to pay $100 every week for this drug. It just doesn't always quite seem fair. They have to pay all this money when the initial discovery was made with taxpayer money. That's the issue that I was getting at. I think I saw one more question, and we'll take a break. Yeah.
Yeah, mine's just, and I think you probably answered it. If you could give an example of a research university that uses public money, like University of Iowa.
Yes, and what's an example of a public research institution? Any of your state universities, and even Grinnell, private university, does receive some federal money for some of its grants. So there's all kinds of research that's being done with taxpayer money. Great, well thanks for your attention. We'll come back after a break and talk about patenting.
Thank you, Josh. We'll be back in 10 minutes. Listen for the...
So you go ahead and come on in as you get your coffee and tea and whatever, and grab a seat, and we'll go ahead and get started again here. While people are coming in, I'll mention something. Someone mentioned to me out in the hallway that when I earned my PhD, that was funded in large part through taxpayer money. So the grants that I got doing research and some of the grants I supported my salary during my training, a lot of it was taxpayer money. And so that's just another example of not only are ideas and targets being supported by taxpayer money, But the workforce, who goes and works for drug companies? PhD students who get their PhD, they go work at a drug company. So the training of the drug company's workforce is also being subsidized by government funding. And so I think that's what gets a lot of people's ire up when they're thinking about how much money drug companies are making and how much of that comes from taxpayer-funded money. It can make people upset. Right, understandably so. So I think there needs to be another solution, and we're going to talk about that a little bit more when we talk about patenting here. So patents, you've probably all heard drug under patent. Let's talk about patenting a little bit. So a patent is essentially a government-sanctioned monopoly on the production and sale of a drug. And patents can vary by country. In the United States, it's generally 20 years is the length of a patent. These can be extended by different things, but that's the rough number. Now keep in mind, as the question was asked earlier, a company might, if they design a new chemical, maybe they don't have a use for it yet, but they don't want someone else to come in and scoop the patent away from them. They might patent a chemical before it has any use yet. So the clock starts ticking. Maybe it sits in the freezer 2, 3, 4 years. Now they find a use for it. It goes through 10 years of drug development. So now 15 years of the patent has expired before it even gets on the market. And they only have 5 years left to make money off of it. So the 20 years is the length of the patent, not how long they have individual sole selling rights with the drug. Now what's the point of a patent? The patent is intended to promote innovation. If I'm going to spend a bunch of money developing a new thing, and as soon as I start selling it, someone comes and looks at it and says, well, I can make that. And then they make it and sell it for a lot cheaper, and they had none of the cost associated with designing the thing. There's not much incentive for someone to go out on a limb and invent something. So patents were designed to say, okay, if you do the work, the expense of inventing something, you should have a while where only you can sell it and make money. The other benefit of this is when you file a patent, you have to describe the thing very clearly. So once the patent ends, now its design is publicly available information. So you get a monopoly for such amount of time, but as soon as that monopoly is over, anyone can make it and sell it. So there's the trade-off there. But this is supposed to be a good. And I think in a large part it is, but I think it's reasonable to ask, I'll come back to this in a second, but I started the sentence. It's reasonable to ask, does it make sense to patent something like a medicine? Okay. So to patent something, the drug, the invention has to be new. No one can patent aspirin because it was known of before patents were invented. So no one can, so it has to be something new that you invented, has to be non-obvious. So you can't take something that already is patented, make a slight modification to it that anyone could have thought of, and then repatent it as a new thing. It has to be a non-obvious item. Or I can't patent like air. No one's patented air yet, but it's really important, right? It's an obvious thing. And it has to be useful. So upon expiration of the patent, generics can be produced. So that's we're going to this information. This is what the drug is. This is its formula. This is how it's made. So after the patent expires, a drug company can come in, scoop in, start making that drug. All they have to do is show that it's exactly the same thing. It works in the same way, so they don't have to do all the clinical trials again. So they have much less expense associated with developing that generic and can be sold at a much lower cost. And then now the drug company who originally sold that, they're going to have to lower their, and then it competes away for both major profits.
What percentage of patents get renewed, and how difficult is that?
To renew it. So the question is, what percentage of patents are renewed and how difficult is it? I don't know. I don't think a lot of patents are renewed.
You can't renew.
Well, you can't renew a patent. There's something called exclusivity, which is related to a patent. It gives you exclusive rights, but it's not exactly the same thing as a patent. That's more negotiable, and that can be extended. And I don't really understand all that legal mumbo jumbo.
Can't you get a patent extension?
If you can get a slight extension, if let's say like the patent office took 5 years to decide whether or not they were going to give you the patent. And now someone else is sort of, there's a way that you can extend the patent for a little bit longer than 20 years, but it doesn't always, it's not, I don't know how common it is. I don't know all that, those sort of legal questions about the patent. So someone can make a generic, show us the same as the drug, start selling it. So the problems with this system. One is people have found ways to get monopolistic control of drugs without the patent. An example of this is EpiPen. So sure, EpiPen's off patent. Anyone could, in theory now, manufacture and sell it, but that doesn't mean that there's still not costs involved with manufacturing and selling it. But just because any company could, not every company does. And so maybe only 1 or 2 companies actually sell the EpiPen and make it. Now you have some investment person come in and says, oh, I see these 3 small companies make the EpiPen, they're the only ones making it, I'll buy all 3 of them. Now I own everyone that makes the EpiPen. Now I have a monopoly. I can start jacking up the price because there's no competition. And the only way for there to be competition would be another drug company would have to decide, oh, I'm going to start making the EpiPen, which might take a few years for them to ramp that up. So that's a problem where, that's just one example, there's other examples of this. So this is an example of where people have had monopolies without a patent. And the question, do patents make ethical sense in medicine? The patents were, I don't think, when they were originally designed, intended for something like medicine. They were thinking more, inventions, like a new dishwasher or something, not a medicine that could have life or death consequences. If you develop a drug, that's the only drug to treat Ebola, and people are dying, and they have to pay $1,000 for your drug because it's the only one that exists, should a patent support that sort of thing? That's the question. So I don't think patents are really intended for something like health or life or death situations, sort of these public good health ideas. Now, a new washing machine, if you don't have the new latest, greatest invented washing machine, your life's not at risk necessarily. I don't think. So those are some of the ethical concerns with drug development. We'll come back to drug companies again, if we have time. I don't know, I'm talking a little bit slower today, so I might have to wait for them next week, a little cliffhanger. But this will all wrap back up. I want to move on to the public service announcement portion of my lecture, a little bit of information about over-the-counter medications. So what's that mean, over-the-counter versus a prescription drug? So an over-the-counter medicine, again, my data's getting a little bit old here. In 2010, which is almost 10 years ago now, gosh, Americans spent an estimated $17 billion on over 100,000 different over-the-counter medications. There's a lot of OTC meds out there being used. And this is sort of slowing down now because we've done a lot of this work, but I think maybe in the late 70s, early 80s, the FDA started this sort of systematic review of over-the-counter medications. Most of you are probably old enough to remember that a generation ago, 30, 40 years ago, there were a lot fewer over-the-counter medications than there are now. This is because the FDA started going through and saying, well, actually, these drugs are pretty safe to use. Why don't we get rid of the prescription and make it easier? So they review, decide, okay, a lot of these things are safe, let's convert them, let's switch them into the OTC category. They also started removing things that weren't really effective or safe. An example could be like sleeping aids. Some sleeping aids used to be made with this drug called an anti-muscarinic. Doesn't really matter what it is. There was theories of why that might help with the sleeping, but it had all these other potential side effects, and it wasn't really that useful in aiding sleeping. So the FDA started removing things like that. It sort of made sense that it would be in a sleeping pill, but wasn't really that effective. So now the consequence of that is now we have a lot more over-the-counter drugs available. And the idea is that an over-the-counter drug would be useful for a condition that someone's able to sort of self-diagnose. I have a cold, someone's able to decide that on their own. And it's simple enough and safe enough to use that they can use it without the doctor telling them how to use it. But doctors can still suggest that you use an over-the-counter medication. They don't have to tell you to use a prescription. So it's important for clinicians to understand these options because there are many safe and effective OTC drugs. Problems, too, can occur with OTC. Even though they're safer, it doesn't mean they're no risk. So there's a lot of extra ingredients that go into these things that can create problems. I'll give some examples of that. And some over-the-counter medications can be misused or abused. So, just some examples of extra ingredients. Here's a list of drugs that are pretty common that people just use, especially this time of year, colds and flu sort of medications. So, Theraflu nighttime has 10% alcohol in it. If you're taking another medication that shouldn't be taken with alcohol, you probably need to know that. Advil PM. Advil is a ibuprofen is the main, but also has antihistamines in there. People might not be aware of that. Pepto-Bismol, Alka-Seltzer have aspirin. Excedrin, extra strength, has 65 milligrams of caffeine. And just for reference, a can of Coke has like 35 milligrams of caffeine. So there's a lot of caffeine in there, which has a medicinal purpose, but someone might not realize that when they're taking that. Local anesthetics like lidocaine that the dentists use, those are in these Sucrets, sore throat. There's a lot of sodium in Alka-Seltzer cold. Advil cold and sinus has these sympathomimetics. These are like decongestions, but they have a lot of side effects that we'll get to in a second. So there's a lot of other stuff that are in these OTCs that a doctor should be aware, oh, I'm prescribing you this, but I should ask, are you taking any other medications over the counter? Because they may have problems with the drug that I'm giving you. As I said, over-the-counter is safe, it doesn't mean no risk. So here's NyQuil, cold and flu. Maybe some of you have taken this before. There's a lot of stuff in there. Acetaminophen is a pain reliever, but it can have liver toxicity depending on things. Dextromethorphan is an antitussive, means anti-cough. Can be a hallucinogenic, some people have used it for that. It's generally pretty safe, but it could have a bad reaction with acetaminophen. So here it is in the same drug. And if you also didn't realize acetaminophen was in here and also took Tylenol because you had a headache while you were taking NyQuil, now you have a lot more acetaminophen at the same time than was intended. A lot of these cold medicines have, or they used to have phenylpropanolamine, a decongestant, caused a lot of hemorrhagic strokes, so it's been removed from the market. It's really similar to pseudoephedrine. These are the same kind of thing. Pseudoephedrine is really highly regulated now. You can only buy so much in a certain amount of time because people use it to make meth. So these are all things that are in over-the-counter drugs that are deemed safe for you to use, but doesn't mean no risk. Another point I wanted to swing back to based on some questions I had, I mentioned way back when vitamin D might have some toxicities. And I showed you this curve and the amount that it takes to be effective and the amount that it takes to be toxic. What I didn't explain very well was toxic doesn't mean, I'm going to die, right? Toxic could be nausea, headache, or something. So what the toxicity is, or what the side effect that there is, each one is measured independently. So maybe it takes 2 or 3 times the effective dose to cause headaches, but it would take 20 times the effective dose to cause something really serious. So just because I said there's a level that could be toxic, it doesn't, there's a range of what toxic means, toxic side effect. Now, the side effects are, can be sort of tricky. So let's take something like Benadryl, which a lot of people might take for allergy medications. These are antihistamines. And remember how we talked about these signaling molecules in the body will have a receptor that they bind to because they do normal things in the body. Well, there's, I think, 5 types of histamine receptor all over the body. Histamine does a lot of different things in your body. So there's H1, H2, H3, H4, H5. There could be what we call on-target side effects of antihistamine. So an example, diphenhydramine, which is used in a lot of these allergy medications, is intended to act on the H1, the number one version histamine receptor. When it acts in certain parts of your body, it does what you want, reduces the allergy effects. However, there are these same H1 receptors in the brain, and when those get blocked, they cause drowsiness. So that's why people get sleepy when they take antihistamines. So, this is an on-target effect. It's a side effect. It's an unintended effect. We don't want people taking allergy medications getting sleepy, but it's not because it's doing the wrong thing. It's hitting what it's intended to target. It's just that target does a lot of things in the body. This is a side effect that's on target. Now, so we mentioned also the brain has this special blood-brain barrier, makes it a little bit harder for things to get into the brain. Well, people knew this, and they said, well, maybe if we can make a drug that still binds these H1 receptors, but can't get into the brain, it'll still treat the allergy, but won't cause a drowsiness. So chemists started fiddling around with these things. Can we change it so it won't get through into the brain, but it'll still bind those H1s in the other part of the body? So they tried this, and they had success with doing that. There's this drug, terfenadine. So this is a second-generation antihistamine. Doesn't get past the brain, which is good. Still blocks the H1, so it still blocks your allergies. That's good. The problem is the change that they made to prevent it from getting across the brain bound to a different potassium channel in your heart, and it was giving people heart attacks. So, that is an example of an off-target effect. It was binding something different than intended and causing the side effect. So there's on-target, off-target, and there's different drowsiness versus a heart attack. Those are both side effects, but they have different consequences. So this should hopefully explain over-the-counter drugs, even though they're safe, can be pretty confusing. For example, there are many different options for the same condition. So there's over 100 or so analgesics. Those are drugs that are used for general pain. There's over 100 or so of those on the market. What's the difference between those 100 different analgesics on the market? Nearly all of them are aspirin, acetaminophen, or some other NSAID like ibuprofen or naproxen. There's only 4 or 5 different chemicals, but they're sold in 100 different versions on the market. Some of these have, as I mentioned, caffeine or antihistamines along for the ride. So this one's ibuprofen, this one's ibuprofen with antihistamine. So there's all this variation that can get pretty confusing. Then you add on top of that, well, you take the same thing, you can have an extended release or gel tab or just pill form. Again, it's the same thing. There's subtle differences. You start to wonder, which one of these things should I take? I just want to help my sore elbow, right? Another confusing problem, extending the brand name. Has anyone heard of Dulcolax?
Yeah.
I use this example, but I don't know if a lot of times my students aren't familiar with this. So there's Dulcolax. There's Dulcolax laxative. Dulcolax laxative, Dulcolax stool softener, Dulcolax balance. If you look at these boxes, they don't look all that different, but there's 3 different active ingredients, 3 different chemicals in these, but they all go by the name Dulcolax, which could get, again, very confusing. If you say, oh, it takes some Dulcolax, well, do you mean laxative, stool softener, or balance? 3 completely different chemicals. So, general guidelines for over-the-counter use. Pick a medicine that treats the symptoms based on the ingredient, not the brand name. So rather than saying Advil, I should get used to saying ibuprofen, for example. Avoid gimmicky stuff if you can. So choose the simplest thing. If you want ibuprofen, buy ibuprofen. Don't buy some version that has ibuprofen and an antihistamine or and a caffeine in it. So buy the simplest version you can find. And particularly for children, very careful attention needs to be paid to dosage form. Is it a pill that they can't swallow? Is it a drink? Palatability is particularly important for children as well. So those are some things where these sort of added things, okay, you got the key active ingredient, but now there are these other formulation concerns that are important for children. So these are general guidelines. Even though you're taking something that's you can just get off the shelf in Walmart or wherever, you still need to pay attention to what you're buying. I'll pause for a second for questions because I have a big new idea that I'm not going to get through. So we'll see if there's any questions right now before we move on. Yes.
There are so many commercials on television for various drugs. Or for over-the-counter stuff. Who is producing over-the-counter drugs? Are they pharmaceutical companies or?
Right, okay. So the question is, there's a lot of commercials for over-the-counter drugs. There's a lot of money being spent on selling them. Who's making them and who's selling them? Yes, those are still drug companies. So a drug company will have sort of a whole profile. We'll have, oh, there's this drug that we made for a long time. We know how to make it. We can make it really cheaply. We sell that to kind of keep the money coming in. We're also doing research on these other things. Or we have the new drug that's the big money maker. Low cholesterol lowering drug, but they still have this profile of other drugs. Some other companies are specialized in generics, so that's their gig. Once a company goes off, once a drug goes off patent, they come in and start making it and selling them. So there's a whole different types. Okay, so I'm going to start on this idea. We'll come back to it and finish it next week, and I think it will fit in nicely with the last topic I had planned. So I wanted to take a little time, we talked mostly about general stuff, to look at a little bit more closely at one category drug. That would be the most popular drug on the planet, the willow tree. No, it's not the willow tree or willow bark intended as a tea. It's aspirin, which is derived as an extract from the bark of the willow tree. So aspirin, plant extracts of aspirin, so salicylate is the chemical name of the main part of aspirin that's functional. It's been used for centuries, maybe millennia, as to reduce pain and reduce fever. So people notice at some point, if you make a tea from the bark of this tree, it reduces fever, reduces pain. So this has been known for a long time. In the 1890s, there was a lot of really good chemistry being done in Germany in the late 1800s. It used to be all sort of chemistry was published in German. That was the language, because that's where all the chemistry work was being done. Felix Hoffmann developed an acetylated version of this. So they took this salicylate, which comes out of the willow, had some problems, really harsh on the stomach, made a slight version to it, added an acetyl group to it at Bayer Laboratories, creating what we know today as aspirin. So this is the most widely used therapeutic drug. There's a really interesting history behind this. I wish I had time to go into it. If you have time to do some Googling and read about Felix Hoffmann and the invention of aspirin, it's kind of interesting. Who gets credit, that sort of thing. So here is your salicylate. Same thing over here, but this little group on the side is the acetyl group. So that's the difference between what you find in a tree and what was made by Felix Hoffmann at Bayer. Why do people take aspirin? Analgesic, reduce pain. Antipyretic, reduce fevers. Anti-inflammatory. Antithrombic, so that's blood thinner. A lot of times baby aspirin is prescribed to older people to reduce the risk of heart attacks. We're going to focus a little bit on anti-inflammatory, but they're all related. All those different things I listed all kind of happen through the same mechanism. So we're going to think a little bit, in order to understand how aspirin and other NSAIDs work, we're going to think a little bit first about what is inflammation and how does inflammation work. So inflammation is a natural response in the body. It's part of your immune system. And it's a natural response to some sort of offending stimulus. It could be mechanical, like you got a cut, exposure to some chemical that's caustic, heat. It's like a sunburn is actually an inflammatory response because your skin was stimulated by the heat from the sun or other chemical or UV rays. Bacterial, other sort of insults. So any sort of offending stimulus to the body will induce inflammation. This is a little grainy, but it's the simplest. I think it's a good diagram for this. This is, in a nutshell, of what's going on in inflammation. So what we have here is, let's say, a blood vessel. These pink, these are the cells lining that. You have the stuff in the blood here. And this would just be kind of the tissue and the fluids around the blood vessel. This little tic-tac looking thing here is supposed to be a bacteria. So let's say you got a bacteria in there. Maybe you got a cut, some bacteria enter the bloodstream and other floating around in the area of the cut. Well, the bacteria recognizes foreign, some sort of offending stimulus, and there are cells, immune cells, their job is to kind of go around and make sure we don't have any of these offending stimuli in our body. And they notice that there's bacteria there. These cells secrete these chemicals, cytokines, chemokines. Those are just words, they're chemicals that are secreted that start the inflammation process. Those chemicals have a variety of effects. One of the main things is they cause the blood vessels to become leakier. So when we talked about capillaries, there's different versions. Some are tighter, some have more holes in them, some are leakier than others. Well, even though they have sort of built-in different levels of leakiness, they're all modifiable, so that they can be made more or less leaky. So the initial response to inflammation is to secrete these chemicals that cause the nearby blood vessel to become leaky. So blood can come out, proteins that are in the blood can come out, cells that can come out. I don't know if you noticed, it's really subtle, but this area went from yellow to pink. So blood is coming out. This is what causes the cardinal signs of inflammation. Redness, heat, swelling, because more blood is coming out of the blood and pooling up in the area where there was infection. So in response to that, these chemicals, more immune cells are brought in, white blood cells, to help fight off the bacteria. The holes also allow those cells to go into the bloodstream and fight the bacteria there. Some more chemicals are released. Some of those signal pain. Pain is also caused by the swelling, creates pressure. Eventually, these white blood cells also secrete chemicals that will recruit other cells that sort of repair the damage. So if it was a cut that was the initial cause of this inflammation, there's sort of some breakdown of the dead tissue and some repairing of that tissue to repair the cut. So all of that is the normal inflammation process. That's supposed to happen. It's a normal part of your immune system. Let's set up this idea, then we'll stop for questions. So inflammation, though, can be good or bad. There's a whole bunch of cells that release a whole bunch of chemicals that cause these signs of inflammation. We just went over redness, swelling, heat, itching. But ultimately, this is supposed to help. This is supposed to be good, to help repair, clean up the infection, and help repair the tissue. So acute, short-term inflammation is good. It's a part of healing. Chronic inflammation that sticks around for longer than it should, is bad, and it's an underlying cause of a lot of different pathologies. So things like, we're going to go over these, but rheumatoid arthritis, gout, asthma, cancer, cardiovascular disease, atherosclerosis, anyone's heard of those terms? A lot of those have inflammation as the underlying cause of them. So for And this is what aspirin and all these NSAIDs target. So next week we'll start up there. We'll talk about a little bit about how inflammation is signaled, how does aspirin and other NSAIDs work to block inflammation, what are some of the consequences of that. And I was going to come back to the drug company thing because there's has anyone heard of like Celebrex or Vioxx? So those were things that were invented, there are anti-inflammatories that are invented for a very specific reason, but then they end up having sort of bad side effects that we'll get into. So, and there's some nefarious things going on with drug companies related to those. So that's what you have to look forward to next week. We have a few minutes. I'll take any other questions people have right now about what we talked about today, if there are any. Yeah.
Yeah, it's right up on your chin.
Working out?
And then
Right
on
here?
the top.
Okay. I think most of the people in this room will be able to relate to this. When I was in high school, biology, I'm going back to personalized medicine.
Okay.
The biology teacher was talking about, oh, there's this new thing called DNA. Okay, that's who you're talking to here.
Okay.
Do you think in your children's lifetime, personalized medicine will be a reality?
Everyone here, microphone works
Yeah.
on that? Okay. So, I don't think it will be in the ideal, ultimate way that people envision it. I mean, the ideal would be you come into the doctor, they maybe swab your cheek cells, an hour later, maybe let's say the next day, they have all your DNA and they know all the drugs and how they're going to interact with you. I think that would be sort of like the goal that people are aiming towards. That's not going to happen for a variety of reasons. A large part just because a lot of diseases we just don't fully understand the genetic basis for them yet. So, I mean, it's already moving that way. People can do genetic screens to see their susceptibility for breast cancer, for example. But that only has so much predictive power. What do you do with information that says you're 50% more likely to get breast cancer? How do you respond to that? So these are some of the questions and the practicalities that are going to limit the benefit of genetics. And I don't think we're going to get a lot more useful in my children's lifetime. I could be surprised. Other questions? One other thing I'll mention to that, since there aren't other questions, since I have a captive audience for 2 more minutes. There are other concerns about the genetics, so let me just think of an example here. So let's say we do get to a point where we can do things like screen and say you have a 40% chance of a heart attack before you're 60. So it doesn't take that much imagination to imagine, let's say someday in the future, my kids' kids, my grandkids going to the doctor says, all right, we're going to treat you for X, let's do the routine DNA screen, so now they have your DNA on file. And somewhere buried in that DNA says you have a 40% chance of a heart attack before you're 60. What do you do with that? We just go on living your life. Now let's say my granddaughter grows up, she's in the Naval Air Force, she flies for them, she retires from there, and now she wants to be a pilot for Southwest or something. She goes to apply for the job and they say, oh, you have a 40% risk of heart attack. We don't want our pilots having a heart attack. Sorry, you can't have the job. So it doesn't take a lot of imagination to imagine how this genetic information while it can be used for good, there could be some problems with that. And one good thing that George Bush, whatever the second, did, there were some laws put in place regarding how this genetic information can be used. So insurance companies aren't supposed to be able to mine your genetic information and not give you insurance because of some risk that you have. But there's a whole can of worms that could be opened with this. So that's another thing that's probably going to slow down the embracement of pharmacogenetics.
Thank you so much.